Hey everyone. It is an honor to have Dr. Emma Lundberg with us today. So Dr. Emma was a professor in cell biology of proteomics at KTH, Royal Institute of Technology in Sweden. And earlier this year, she recently moved to Stanford. And as we hear, the weather is really nice there. And so now she's an associate professor at the Department of Bioengineering at Stanford. And her research groups really works to understand how variations and deviations in protein expression patterns can contribute to cellular function and disease. And she also directs the Cell Atlas wing of the Human Protein Atlas program, where she has been leading a major effort for over 10 years now to create the subcellular map of the entire human proteome. And we all know Emma has been recipient of many awards, and most recently she won the yoran Gustafson Prize, if I'm pronouncing that correct, from the Swedish Academy of Sciences for her pioneering work in developing technologies and tools for really large-scale characterization of the human proteome. So without further ado, Emma, we are very happy to have you here, and I very much look forward to your talk.
Thank you so much, and thanks for inviting me. And you have great Swedish pronunciation, I have to say also. That's not an easy name to pronounce. Yes, so I'm going to talk to you a bit about our work to understand the spatiotemporal architecture, proteome architecture of human cells. And as you all know, this is a special forum where everyone thinks about space, I guess, and spatial distribution of molecules. Biological processes are partitioned in time and space, and so are also proteins. And we also know that relocalization of proteins is an important form of functional regulation, and mislocalization may be a cause of disease. So in my opinion, it's very important that when we try to understand cells as system, we also have to map where the molecules are, where the proteins are. That's what my lab is interested in. Can I just ask you, do you see my full screen or the presenter's view?
We see the full screen.
Full screen, good, great. And my work is closely connected to the Human Protein Atlas. As you heard, I'm one of the co-directors here. I'm not going to go into any of the details. We could talk about antibody validation for an hour probably, but I just wanted to say that we've generated antibodies for the majority of human proteins, and I'm happy to answer any questions if you have questions about how we generate this data or the reagents that we have or how we validate it. But please do make use of this database. It's a very rich resource with data. In particular, image-based data. So we have about 20 million images that have been systematically acquired and systematically annotated, either for protein distribution in tissues using immunohistochemistry or protein distribution at the subcellular level in cultivated cell lines using immunofluorescence. And well, it's a great image collection for building machine learning models, which I'll get back to later as well. So, but today I'm going to focus on mainly the subcellular distribution of proteins. And for this purpose, we wanted to have an automated pipeline so that we can localize proteins with high resolution. And we decided to go with just regular oil immersion confocal microscopy that we automated ourselves, and also automated the sample preparation pipeline. So we have DOPI for the nucleus. We have antibody-based markers for microtubules and the endoplasmic reticulum, both to be able to quality approve the fixation, but also for computational purposes and getting a nice outline of the cell. And on top of this, we add HPA antibodies to systematically localize human proteins one by one. Here we see CD44, and we can conclude that it's on the surface of this cell. Do you see my pointer? Yeah, good. So in this talk, I will mainly show the HPA antibody and the protein of interest in green. So I hope you can see that. If it looks like this, we'll conclude that it's actin filaments, or protein is in nuclear speckles, or in centrosomes, for example. So this is the work that we've done for over a decade, and a couple of years back, we published the first subcellular map of the human proteome, where we had localized, I think now we're up to 14,000 proteins to 34 different cellular structures. And that's more or less what is expressed in cultivated cell lines. And also, it's hard to localize secreted proteins, so we have to focus on the intracellular proteins. We could define organelle proteomes, and still today my lab is focusing a lot on this image collection and using it as a basis for further studies. So that's what I'm going to talk about today. But something that I always want to point out is that We showed that half of all human proteins localize to more than one compartment. You can see that here. This protein is in the nucleus and on the plasma membrane. And this has also been shown for mouse and yeast, and it has more recently also been demonstrated using different technologies such as mass spectrometry. And the number now is even higher. It's somewhere between 55 to 65% of all human proteins localized to multiple compartments. So these multi-localizing proteins may be multifunctional proteins. They may have a context-specific function or moonlight in different parts of the cell. And this then increases the functionality of the proteome and also the complexity of the cell from a systems perspective. So again, that's why I think when we model cells, we cannot do it on a gene kind of basis level. We have to look at where the proteins are as well in order to truly understand the function. And to add to this complexity, in this image, you can see that all the cells looks rather identical. Of course, that's not always the case. This is a cultivated cell line. The cells are more or less genetically identical. They're not synchronized though, but you can see this one protein, enolase 1, it localizes and expresses quite differently in these cells. Sometimes it's in the nucleus, sometimes in the cytosol, sometimes both. We also see it on the plasma membrane in some cells, plus the expression levels vary quite greatly. And in this case, enolase 1 is one of the few well-characterized moonlighting enzyme proteins out there. I think there's 200 proteins that have been described to have multiple functions, and enolase is one of them. So we know it has completely different functions in these three compartments, and then the heterogeneity is just increased by these abundance levels. So by regulating this one protein, the localization and abundance, the cells get very different functional capacities. So we get a great functional heterogeneity by this spatiotemporal proteome remodeling, so to say. And we wanted to understand what's the extent of this spatiotemporal heterogeneity, not only for enolase 1, but for all human proteins. So we've been mining our images, and we can see that about 19% of the human proteome shows signs of heterogeneity, or what we call spatiotemporal variability. It can either be, as you see to the left here, translocation. This protein is in the Golgi apparatus, but when the cell's divided, it's in the endoplasmic reticulum. Or it can be that it's only expressed in a subset of the cells, or it can be that it varies in intensity or abundance between different cells. So based on this, and particularly this image to the left here, we thought that these are still non-synchronized cells. They're in log phase growth. Probably most of these variations just we can attribute them to the cell cycle. So let's test that hypothesis. So we decided to work with the U2S Fuji cell line. So it's a cell line that has two fluorescent indicators that basically a red and a green fluorescent markers that change over the cell cycle. So we can fit a model to the cell cycle trajectory that you can see here. And we can make a linear pseudo-time representation of the cell cycle time. So using this cell line, we sorted the cells and did single cell sequencing after measuring the fluorescence. And we also did co-staining with the 19%, with antibodies for the 19% of the proteome that tended to show spatiotemporal variability. So these are just mock-up plots, but the idea was that if the protein is varying in relation to the cell cycle, we would see some kind of profile, temporal profile occurring. If it's independent of the cell cycle, we would see a flat line and still high variability. And just to skip to the end of this story, at the top here are some real data plots. You can see the data is noisy, but it's still okay. The Fuji cell model is reasonable in terms of noise. So at the top here, you can see BUB1B and cyclin E1, well-known cell cycle regulators. And here we can see a temporal profile both at the protein and RNA level. And these proteins are known to be regulated at the transcript level. But if you look at the bottom here, we see DUSP18 and DUSP19. It's two proteins in the family of dual phosphatases. They have no literature connection to proliferation or the cell cycle, but we can clearly see a temporal profile at the protein level where the protein is accumulating either in G2 or G1. But here we see that the RNA is just stable. It's a flat line, right? So we assume that DUSP18 and DUSP19 are regulated at a translational or even more likely post-translational level. And in total, we were quite surprised. We found 300 new cell cycle proteins, considering how well studied the cell cycle is. Most of them are stable at the RNA level. That's probably why they have not been discovered before. We could also determine that the average temporal delay between RNA and protein, if we look at time of peak expression, is 8.6 hours, which might seem long at first, but it corresponds very well to what has been shown in the circadian rhythm. We find many, of course, interesting proteins, potential new markers for cell proliferation, and we also did some gene silencing to show that these proteins are not only passengers of the cell cycle, they do actually influence growth in different ways. And I also want to point out that it's very common with cell cycle dependent translocations as well, particularly to the nucleus in G2 or later in the cell cycle. And this was published last year in a paper in Nature, and it was a very fun study, but I think the most fun part begins here, because our initial hypothesis that the majority of the variable proteins would map to the cell cycle was wrong. We could only explain 1/3 of the variable proteins by correlation to the cell cycle. So 2/3 do not correlate to the cell cycle. So why do we see such extensive variability? The magnitude, the amplitude of the variability is as great as for the cell cycle dependent ones, but we don't know the biological meaning of it. What we do see is that these proteins are enriched for being in certain subcellular locations. They're enriched for being in the cytosol, in intermediate filaments, and in mitochondria. For example, compared to the cell cycle proteins, they're mainly found in nucleus, nucleolus, and mitotic structures. We also see a great enrichment for metabolic enzymes here. So if we, again, group this together and look at all variable proteins, I told you the number was 19% for the human proteome, but the corresponding number for metabolic enzymes is as high as 40%. And it goes across many types of metabolism. So this is, of course, very interesting. Why do we see so much dynamics when it comes to metabolic enzymes? Again, I'll show you one example. This is HMGCS1, an enzyme involved in cholesterol biosynthesis. And again, you can see the nuclear localization, the cytosolic location. This is where the canonical function happens. There's no literature about its function in the nucleus or even being in the nucleus. And also you can see we have cells with high expression and cells with low expression. So here we don't have a pseudotime to map the data to. We're always a bit concerned about cross-reactivity to the nucleus when it comes to antibodies. So we decided to work with the Chan Zuckerberg Biohub here in San Francisco and tag this cell line together with Manula and Eddie. So we did biallelic GFP tagging, and then we took a single cell and let that cell divide a couple of times. And we wanted to see if this single cell could reproduce all the distinct protein expression phenotypes. And this is the results that we got. So it's pretty obvious that it can. We still see the different localizations. We see high and low abundance. And this was the case for many of the enzymes that we tagged. But then we started thinking, but what if, okay, it's not dependent on the antibodies, it's not an artifact there, but what if it's just an artifact in cell lines and this is not of relevance in tissues? Then maybe we should just turn to the tissue atlas of HPA and confirm that we can see this variability also there. And of course, this is just one example. This is HMGCS1, again, in the glandular cells of stomach and also CNR cells in pancreas. And again, we see the variability of expressing cells, non-expressing cells, nuclear localization, cytosolic localization in cells that are supposed to be identical. So at this point, I'm not going to talk about it today, but we're wrapping up a big study where we've analyzed metabolic enzyme spatial heterogeneity in liver and pancreas. And I can tell you it's extensive and it's very interesting. So at this point, we're still sitting on massive data sets and have many questions. What's the cause and consequence of these spatiotemporal variations? What's the level of cellular regulation? Is it RNA or protein? A lot of it is post-translational, as it seems, where we are now. How do intrinsic and extrinsic factors influence the single cell metabolic state? Can we, for example, hold one cell out and predict the state of that cell given the other cells in the image? And how do metabolic and proliferative states interconnect? I didn't really talk about that, but what we do see quite often is metabolic enzymes translocating to the nucleus. It might not always happen, but when it happens at certain time points of the cell cycle. So I think there's an interesting interconnection here that we can explore further. So these are things that my lab here at Stanford will be working more on going forward. But again, everything that I've described so far is kind of critically dependent on that we can classify patterns in images and quantify the information that we have here. And it's a lot easier with cell lines than tissues that I know a lot of you are working with, but it's still not trivial. So we have, of course, great class imbalance. Some patterns are common, some patterns are rare. We work with 60 different cell lines. They look very different. Here you see the same actin filament protein in 3 different cell lines. So we need robust generalizable models. Then we have the single cell variations. So ideally, we want models that not only labels the image, but actually labels every cell in the image. And then we have multi-localizing proteins. So we need models that can see if it's one or several labels and maybe even unmixed patterns. And since I'm working with the Human Protein Atlas project, we tend to do things to engage our users. And over the years, we've done several different crowdsourced efforts to improve pattern classification schemes. First, we did a citizen science project where we tapped into a computer game and had 300,000 gamers help us. And then we've hosted 2 Kaggle competitions. The first one was just to basically beat the gamers, develop machine learning models to classify the patterns that you see in a full image. The bottom one there, the recent Kaggle competition that closed earlier this year. It was a competition to provide labels for every single cell in an image, but given only weak, noisy input for the training data. And both challenges have been super successful. It's a very fun way. You have to work hard to set up the competition and think about how to do it so that the outcome is good. But in the end, it's a very good way of serving many models in a short time. So today I will briefly show results from both point 3 and 2 here. So first, I just want to say we're just wrapping up the paper for the second Kaggle competition, but it seems to be working very, very well. And I just want to show you these class activation maps. So here we use gradient weighted class activation maps to visualize basically the regions of the images that the model looks at. And these are single cells that we cropped from images. And you can see that it's mixed patterns. In this top cell, we have the mitochondria and the nucleus. Here as well, we have even nucleoplasm and nuclear membrane stained here. And what is cool is that you can see that the model really looks at the biologically relevant regions when it's classifying the corresponding patterns, even for these mixed patterns. So by now, we're very confident that these models are looking at biologically relevant features and that we can use them to really start to dissect the spatiotemporal variability of the human protein across cells of different shapes and forms and other things. So this opens up for a lot of, I would say, single cell modeling of proteome shape. And this is, if we use these models, now I'm showing you the data from the first Kaggle competition, but they basically look the same. It looks a bit better from the second one. But if we use this model that was developed to classify the patterns in the images as a feature extractor, so we just take the features from the penultimate layer and then we reduce the dimensions here into a UMAP plot. And what you see here is all images in the HPA cell atlas. So this is about 80,000 images corresponding to 13,000 proteins mapped across a wide variety of cell lines. We've colored each image according to its manual annotations that we provided. So you can clearly see that the model nicely separates the different locations, but that's what it was trained to do. So that's fine. What is also nice is that we can see that if you think of it, a pattern is a punctate pattern in the nucleus, like a round dot, could be very similar to a round dot in the cytosol. The only difference is where they're located in the cell. So it could be so that the model would group them together because the pattern is very similar. But here we can see that that's actually not the case because these are cytosolic punctate patterns. And here we have nuclear punctate patterns, for example. And actually this entire cloud is nuclear patterns and this is cytosolic patterns. So thanks to the reference markers, this model is not only just classifying the pattern, but it's actually looking at where the pattern is in relation to the landmarks. And that means that this model is kind of embedding the spatial distribution of the protein in the cell, not only just classifying the pattern, if you see what I mean. And this is very important because this made us think that this must be very suitable for modeling the spatial distribution of proteins in cells. So we were very happy with this. A second thing that we were also very happy with is all these gray dots. They represent multi-localizing proteins. And if you look at, for example, F or D here, they're multi-localizing proteins. F is in the nucleus and nucleoli, and we can see that it ends up right in between the pure clusters of nucleoli and nucleus. It seems like this model also handles multi-localizing proteins in a nice way. So then we started thinking, so what can we do with this? How can we model cells? What is spatial distribution in cells anyway? Ultimately, what do we want to do? We want to understand cell function. But what is cell function, right? Well, cells are, as you know, hierarchical systems organized into different scales, and different biological functions will be exerted at different scales. And we're only mapping the protein distribution at the organelle level. So we cannot really expect to be able to explain biological function at that level alone. So we were thinking that what if we instead try to build a hierarchical map of this proteome architecture in the cell by integrating our data that is measured here with protein-protein interaction data that is measured here. Basically, global distributions of proteins in cells integrated with local neighborhoods of proteins in cells. Maybe we can then build a multi-scale hierarchy of a cell. And this has been a great collaboration with Professor Trey Eidecker at UCSD and his student, Yue Kin, that led this work. So what we did is that we took the 800 proteins we had mapped in HEC293 cells that were mapped both by our images and by protein-protein APMS data to get protein-protein interactions from the Bioplex database out of Steve Gige's lab at Harvard. And then we embedded this information using our machine learning model or a node-to-vec model. We can concatenate these vectors, and then we can start to measure pairwise protein distances. And what is neat here is that we could also, in images, we were used to working with distances, but protein-protein interaction data is rarely calibrated to scale. But here we could make use of the fact that we do have scale in images and that there is a lot of measurements in scale of the literature. So we could actually calibrate these pairwise feature vector distances to actual distance in nanometer. And then we could build this multi-scale subcellular hierarchy of the cells. So basically we're looking for communities in the data that are at the bottom here, very stringent, like tight complexes, and then gradually we relax the parameters and find larger and larger systems in the cell. And at the top, of course, this would be like the parent, the cell node. And this here we revealed 69 cellular systems, and it kind of resembles a gene ontology that you're used to seeing, but this is purely data driven. And we see the cytoplasm and the nucleus, and then we see smaller and smaller and smaller systems. Of course, this is only a small part of the cell, but what is interesting is that half of these systems were already represented in gene ontology, half of them were new. So we spent, I think, a year just generating validation data to prove that these systems are indeed real functional systems in the cell. And we could, for example, reveal crosstalk between cytoplasmic and mitochondrial ribosomes and identify new roles for poorly characterized proteins and so on. And I think what makes this approach powerful is that we call this map MUSIC, multi-scale integrated cell, is that it captures the pleiotropic effects of the multi-localizing proteins and gives protein-protein interactions a spatial dimension. And I also think it's an interesting framework for thinking about how to incorporate other types of molecular data or look at other types of interaction data. And I do want to show this little plot from the supplementary, but I think it's just nice.
Give me 1 second. I'll send this out.
Oh, okay, sorry. Someone is not muted. If we try to build this map on images alone, we only get 2 or 3 systems. On PPI data alone, we only get 10, 11 systems. But when we integrate the data, that's when we get the 69 different systems. So the power really comes with this integration across scale. And then I will just spend a few minutes, and then I know I'm supposed to end. But ultimately, the music map is very cool. It's very fun, but we're used to working with images. And I think we're also losing a lot of information when we're aggregating it into this music framework. Ideally, we want to, work with the morphological scaffolds and the shape information that we have in images. So a lot of our modeling work is now focused on different ways to basically generate functional maps like the music map on a given morphological scaffold, a given shape. So we can also start to explore proteome distribution in relation to shape. And the ultimate aim would be to build a whole proteome model that would allow perturbations of spatiotemporal distribution of proteins and prediction of function. But that's, of course, far down the line. But we were thinking, what's the power of our data? We only have single sections. We don't have any dynamic data, but we have a lot of images. And what we can do well with a lot of images is, of course, to train models to generate fake images like style GANs or diffusion models now, where you can generate faces and tune them and say add glasses, remove glasses, change hair, and so on. So we trained a model to generate fake HPA style images. So at the top here, you can see fake images. At the bottom, you can see real images. So we can tell the model to generate a fake image of this cell line, and we want the cells to be this confluent or this sparse, and so on. And of course, this is still fake cells, or it's a representation of a cell based on data. And the plan is to go from this kind of pixel-based predictions to transform these pixels into objects, because then we can start to add biophysical restraints to the objects, and we can start to add conditions, knowing that this protein should interact with this protein. And then we can, based on this model, hopefully go towards this super multiplexed image with entire proteome, or at least being able to project music-like models onto a more morphological or shape framework. So we call it our vision of a whole proteome digital cell, and it's still a sandbox and we're playing with it, but it's great fun, and I think it will evolve quite a lot in the near future. And with that, I'd like to end my talk and also say that we do work a lot with highly multiplexed imaging, of course, because to really understand these spatiotemporal variations, we need to work in a tissue or organoid context, and we need to look at more than 1 or 2 or 3 proteins at a time. So, but I didn't show that in my talk today in interest of time. But thank you so much, and we'll always continue to promote open science and share labeled and raw data and code freely and widely. And I'd like to thank my group. They're really amazing. A lot of them are coming with me here to Stanford, so that's great. And thank also funders and you for listening. I'm happy to take questions.
Thank you very much, Emma. That was spectacular. Are there any questions? Probably I can start with a question. I really like the way you kind of describe the spatial communities in proteins within a cell. As you mentioned, like this is going to be really important, as that could serve as a framework of how we think about communities as cells, and then that forms a tissue and then into an organ. I was wondering, like, what was the prior knowledge that you used when you talked about protein to protein interactions?
Yeah, so we basically didn't use any, well, we didn't use any prior knowledge except in the calibration to scale step. Mm-hmm. So protein-protein interaction was data from Bioplex, the images was our data, but in that calibration, we reasoned that there is a lot of knowledge about the size of different systems, and there's also a lot of knowledge about the number of proteins in different systems. And it turns out that we can actually generate a pretty accurate calibration function in between. It's not perfect, but it's doable. So that was the only prior knowledge that we added.
That's nice. Ken?
Oh, Emma, I'm just wondering, do you have any thoughts about RNA protein spatial colocalization?
Yes. Yes, I don't have any data to back it up, but I'm pretty sure that there is a correlation. I'm pretty sure that RNAs that are closer to each other, if you look at a pair of RNA, pairwise comparison, for example, different transcripts, I'm pretty sure that the proteins would have if the RNA have a similar distributions, the proteins would have a similar distribution. This is, if in prokaryotes, it's very well conserved. I assume it will be conserved to some extent in eukaryotes. I don't know if it's conserved all the way from the DNA, like 3D distances. That would be cool. I don't know if that's the case in the nucleus as well. But I think there are several studies I know that are on the way of being submitted, published, that look into that. And there seem to be some correlations, yes. Then if it's at what level it is, if it's more that, secreted transcripts of secreted proteins tend to be closer to the ER, or transcripts of proteins that should go to the nucleus tend to be closer to the nucleus, that I don't know, but I think it might be more elaborate than that.
Thank you.
But with that comes also an interesting other thought. There's a lot of different proteoforms for ribosomes, and how do they contribute to this localized translation, I think is very interesting as well.
Maren?
Yes, thank you very much, Emma. This was really a beautiful talk. I was wondering, because you mentioned the spatiotemporal heterogeneity of proteins within, meaning the spatial distribution of some proteins may change within the cell cycle of for some proteins. And I wonder if you ever moved up one more level and maybe looked at the life cycle of a cell, or even within, if you look at tissue or organisms, like the age of the organism, going even up another level.
Yes, so we haven't done that. We're actually thinking a lot about that now. And I don't know if you're working with that or people here. Would you know what would be the best cell model system for aging? That's what we're discussing earlier this week. So I'd love to hear it if anyone has any preferences there. Because I think a lot of it, especially these metabolic variations, maybe the cells are more dynamic when they're young, less dynamic when they're old, or I don't know.
No, it's complex. I mean, I just thought of this while you gave this presentation, and of course it's like so heterogeneous, even like within the same organism, but in different organs. And yeah, there's a lot of complexity, but yeah, it's interesting. I just would love to discuss more.
Yeah. So any input there, I'm very happy about. Thank you. Great question. There's a question in the chat also. Oh yeah, Jan, when you're.
We cannot hear you, Yanwen, yet. I'll probably read out the question. So Yanwen asks, I wonder, how do you get the true label for the deep learning models? Do you manually label each cell in an image? And how many images do you need?
Yes, good question. So for the Protein Atlas, we've manually labeled images, all cells in an image. And there we have labels for hundreds of thousands of images. But for the second Kaggle competition, when we wanted to develop models for single cells, we thought it's not reasonable that we would label millions of single cells, right? And also, if we look at models that are available for image classification, most of them are developed at an image level. So we were thinking that it could actually be interesting if people could develop frameworks for how to go from image level labels to precise labels, so that we can better make use of available models. So that's why we designed that competition, so that the participants were given images with image level labels, so noisy data. And then we labeled a smaller subset. We labeled 50,000 cells from 2,000 images approximately, or maybe 10,000 images. And then we labeled every single cell there. And there we chose images where we knew that there was a lot of heterogeneity, but that image set has been kept hidden. So it's all hidden at the Kaggle, so you can use it. Part of it was used for kind of public testing, so they can develop their algorithms and then test it, and they have to trust the leaderboard. And then we had the kind of hidden data set that was used for the final ranking, and it was very well split, and it wasn't a lot of shakeup, so that turned out very well. So there's the data sets are there, and there's at least 40,000 single cells that have been labeled.
So who are these people who labeled 40,000 single cells?
My lab. You want to join my lab? No, it's actually, we have a lot of tools. We develop tools also for, we have a lot of super flexible tools for doing image annotation. So this is not a lot of work, to be honest. We have tools that are super fast, super quick. So we often do, we just annotated a set of images from 50,000 cores of pancreas tissue samples, and that maybe took a couple of weeks, but it's a lot faster because we have these flexible tools that we can use for collaborative annotations.
Are these tools publicly available?
Yeah, they are, but not all are published. So if you want to use them, please reach out. They often end up being hidden somewhere in the supplementary, which is kind of sad, but yeah, reach out and we can show how to use them.
Thank you. I'm sure Clarence is going to ask about similar things. Clarence?
Hi, I'm not, I wasn't going to ask about that at all, but I mean,
Clarence does do a lot of those annotations himself.
I only did 10,000, not 40,000. That's not as many as you. My question was actually more about the kind of image acquisition setup, also the microscopy setup that you had. So is that from like a confocal, because they were very high resolution images that you sent? Okay.
Yeah. Oil immersion. 63x.
Oil immersion. How are those images directly translatable to training another model that maybe you want to apply on like lower resolution images? And if not, would one have to repeat the exercise on lower resolution images?
Yeah, we've actually been thinking about publishing such a data set. It's a very good question. I think it would translate pretty well to, we've used some other resolutions and it translates pretty well. What it doesn't translate well to is if you use different reference markers.
Right. Okay. I see. Is
So
that because
that will be the problem. But there are other, I mean, you can use the framework, you can retrain it maybe with other reference markers or use other self-supervised methods for embedding the spatial information.
sorry, what is the limitation with the reference markers? Do you mean with it?
That these models are trained to also have the microtubule and the ER markers so that it can kind of predict, embed the spatial distribution in relation to those.
Oh, I see.
If you remove one of them, you're pretty good. If you remove both of them, the models will not do that well. Then there are other models that will perform better.
Oh, okay. Thank you.
But would you be interested in a matched data set? If we produced a matched data set of, let's say, 200 proteins with different imaging resolutions, would that be of use for you?
I think so. I mean, anything of that magnitude would be very, very useful. Are you thinking of training like GANs or something to try and bridge the gap between?
No, we were just thinking that since the antibodies, the cells are fixed and our sample plates are actually stable for 6 months. So we've been saying for a long time, we just never got around to it, that we should just take them and image them on all the different systems that we can find, more or less, to have different resolutions, different systems, different as much as we can for, I don't know, a couple of hundred proteins.
Mm-hmm. Yeah, that's right. Yeah, we have a couple of objective lenses. Some are air, some are oil and water. It'd be interesting to actually characterize these systems as well.
Bailey?
Hi, thanks so much, Emma, for the talk. Marin's question got me, a similar question got me thinking about aging and different cell lines, and that could be used as models. A similar question. Sorry. Slightly tangential question. So have you done this, like shown the spatial temporal heterogeneity in any primary cells or purified primary cell lines? Because I'm just wondering if there's any amount that could be like either associated with like just classic genetic drift of these cell lines that you're working with. And then in terms of speaking or thinking about aging cell line models, wondering if you've like considered progeria, or if there's any like reason that would like maybe it not be a model of choice for cell lines.
Yeah, very good question. So we are considering progeria. We're looking into those cell lines now. I think they would work, often fibroblast based, they would work, or iPSC based, they would work very well with our pipeline. So I think that's great. As for genetic drift, we didn't look that much at primary cells, but instead we've looked at the tissues.
Okay.
So I think that we're pretty convinced that it's not genetic drift.
Okay.
And also from the tagging, when we take a single cell and just let it divide for a couple of divisions, it's also not going to be genetic drift. So these fluctuations are much faster. But what is interesting is also the temporal that you often don't think about, but makes it complicated to design good imaging experiments, is that the temporal fluctuations that we see are at different time scales. So for example, we can see that an enzyme goes in and out of the nucleus at like a 4 or 5 hour cycles. But then its abundance levels from high and low changes at a 72-hour time scale, right? So it becomes very hard to design good live cell imaging experiments that can capture all of this data. So we also have to think about time, the dimensions of time here as well.
Got it. Okay, thank you.
And then I see someone has written about C. elegans and Drosophila, that I know they would be great. It's just so sad that our antibodies are generated for human proteins. So for us, it's just a lot of extra work to validate them in other species, even though they often work, but it's not what we prefer.
Do you think a 3-dimensional or the third and y-axis within your data of image acquisition would help with a better resolution of localization, or do you think you're capturing most of the protein localization just on 2D?
We're capturing most, well, actually we do acquire some said sections, and when there's sometimes you cannot capture focal adhesions and nucleoli in the same section with confocal. That's just not going to happen. So then we take several sections to capture that. So I don't think we've missed a lot of locations, but of course, if we do want to model, we're doing, I also didn't show that, we're doing a lot of modeling of the shapes of the cells. And of course, that's limited to the assumption that our sections are representative to the kind of representation of the cells. So I think when we want to move to more modeling of the cell, it would be advantageous with 3D images. On the other hand, we have worked with very flat cell lines. I think we're still good working in 2 dimensions, but if I would have to choose, I would still, for example, choose between getting 3-dimensional image data sets, or I think it's more important to go into better model systems such as organoids where we have a controlled environment as well. I think that will be, I think we're more restricted by the use of cell lines than we are with the lack of 3D images. But for example, Alan's cell are doing great work in terms of organoid or organelle modeling in 3 dimensions. So there are other efforts pursuing that.
Any other questions? Okay, so if not, I'll probably thank Emma once again. Thank you very much for coming. It was a spectacular presentation.